Hello everyone, we are Women's Circle and we are a group of IB higher level biology students. Today in this video, we will talk about A1.2 nucleic acids. In the previous video, we have covered the topics properties of water and origin of water on Earth. Today, we will talk about the structure of nucleic acids, directionality of DNA and RNA strands, purine-to-pyrimidine bonding and packaging of DNA, Hershey-Chase experiment, and lastly, Chargaff's rule. So let's start by talking about structure of nucleic acids. We should firstly understand that DNA is the genetic material of all living organisms. But what is DNA? DNA is the short of deoxyribonucleic acid, and it is long-term stored genetic information for all organisms on Earth. Mutations that happen within DNA pass on to the next generation. DNA is universal to all living organisms, and this proves common ancestry between living organisms. DNA's structural framework is a nucleotide. Many nucleotides join together and form nucleic acids. DNA is a type of nucleic acid. Within the DNA, specific regions of the nucleic acid may code for specific traits, like the eye color. These regions are called as genes. Later on, these genes usually code for amino acids, and amino acids form proteins. Cell's identity and function is determined by the proteins it is able to synthesize, so this point is important. So let's continue by talking about the structure of a nucleotide. Basically, a nucleotide consists of a phosphate group, a 5-carbon monosaccharide pentose sugar, and a nitrogenous base. These three are connected to each other by covalent bonds. There are 4 nitrogenous bases in the DNA structure: adenine, guanine, thymine, and cytosine. But originally, there are 5 types of nitrogenous bases in total, when we include uracil as well. In terms of different pentose sugars, because there are 2 types of pentose sugars, ribose is present in RNA while deoxyribose is present in DNA. Knowing their difference is important for the IB. The ribose includes an OH group attached to its carbon 2, while the deoxyribose includes only hydrogen attached to its carbon 2. The name deoxyribose comes from there as well. Deoxy basically means lacking one oxygen. So their difference is basically that deoxyribose lacks one oxygen attached to its carbon 2, but ribose has that oxygen. Now we should look at the sugar phosphate backbone, which is extremely important for the DNA structure. Nucleotides bond together to produce long chains in polymers, as we said before. But how do they do that? One pentose sugar of a nucleotide is bonded to the phosphate of the lower nucleotide as a result of condensation reaction. As we can see in the drawing, the pentose sugar of a nucleotide here is bonded to the phosphate group of the lower nucleotide. Covalent bonds are formed between them, and these bonds are quite the strong bonds. They are harder to break, which makes the polymer stable. The sugar phosphate backbone, as you can understand, gives the molecule stability, so it is extremely important. Now look at the structure of RNA. Nucleotides join together by condensation reactions between the pentose sugar of one nucleotide and the phosphate group of the next nucleotide. As a result, a water molecule is released alongside with the polymer, like we said on the previous slide. As you can see from the drawing, RNA is a single-stranded structure. It is the genetic material of some viruses, but it is not a universal genetic material. It has uracil in its structure instead of thymine. Now let's look at the DNA structure. The structure of DNA. It is a double helix made of two antiparallel strands of nucleotides. The two strands are linked to each other by complementary base pairing between nitrogenous bases by hydrogen bonds. Complementary base pairing is very important for DNA. It allows DNA replication, and two exact copies can be made because in complementary base pairing, adenine only bonds with thymine and cytosine only bonds with guanine. Because of this specific bonding, DNA knows how to make correct copies of itself. The two strands of DNA are connected to each other by hydrogen bonds. Thymine can make 2 hydrogen bonds with adenine, while cytosine can make 3 hydrogen bonds with guanine. The structure of DNA, as we stated, is antiparallel. As you look at the strands, you will realize that one goes from 5' to 3' and the other goes from 3' to 5'. They are in the anti-direction, but still they are parallel to each other. This makes the structure of DNA antiparallel. Now look at the differences between DNA and RNA. They are both linear structures. They both contain a phosphate group and a pentose sugar. But what are their differences? So the DNA is a double-stranded molecule, while the RNA is a single-stranded molecule. DNA includes deoxyribose pentose sugar, and RNA includes ribose pentose sugar. DNA includes thymine as a nitrogenous base. RNA, on the other hand, includes uracil as a nitrogenous base. DNA has a double helix structure, but RNA varies on shape depending on the type of RNA, which we will talk about in a second. DNA is the permanent genetic code. It is the universal genetic code of all organisms, while RNA is not a permanent genetic code except for some of viruses. Now, let's look at important nucleic acids and types of RNA. We will talk about these in detail in the next chapters, but for now we need to know them and their basic functions. mRNA is messenger RNA. It is synthesized from a gene from the DNA after transcription process. It carries the necessary genetic information to make a protein. tRNA is transfer RNA. It transfers the correct amino acids into a growing chain of amino acids during protein synthesis or specifically in translation. rRNA is ribosomal RNA. rRNA is used to create an organelle called ribosome. Proteins are synthesized in ribosomes. And lastly, we have ATP, adenine triphosphate, single nucleotide nucleic acid. It gives chemical energy to the cell, and it is not used to store energy as it is an unstable molecule. The storing energy is done by macromolecules like carbohydrates. Now we will talk about the HL material of the topic. We will talk about directionality of DNA and RNA strands, pure interpyrimidine bonding, and packaging of DNA. So, directionality of DNA and RNA strands. As we know, 5' to 3' linkages in the phosphate sugar backbone is present in DNA. This refers to the 3rd and 5th carbon atoms in the ribose or deoxyribose. In DNA, one strand runs from 5 to 3', while the other runs from 3' to 5'. This is not because they were synthesized differently, but it's because of the antiparallel structure of DNA. Importance of directionality. When a nucleotide is added to a nucleic acid chain, it is not random. It bonds in a specific way. Directionality allows this order. And it is also important in DNA replication. Two strands are separated. Both act as a template for the new strands that will be synthesized. The new strands will always begin from 5'. So, when the new strands are being coded, DNA's directionality allows correct coding and replication. Another place where the directionality is quite important is during protein synthesis. In protein synthesis, one gene in DNA is opened. From that gene, one RNA molecule is synthesized due to complementary base pairing, starting from the 5' end. That RNA molecule is later turned into mRNA. This process is called as transcription. After that, in translation, mRNA is again read in 5 to 3' end to produce a polypeptide chain at the ribosome. This is done by tRNA, and this process is again called as translation. We will learn about protein synthesis in detail in the later topics, but for now we need to know that 5 to 3' end reading is quite important. Now we have purine to pyrimidine bonding. Nitrogenous bases are grouped into two, purines and pyrimidines. Purines include adenine and guanine, while pyrimidines include cytosine, thymine, and uracil. Their difference is the number of their rings. For example, guanine and adenine are double-ringed structures, as you can see in the drawing, while pyrimidines are single-ringed, like cytosine and thymine here. This bonding allows accuracy during complementary base pairing. It stabilizes the DNA structure, and two strands are always at a consistent distance from each other. Packaging of DNA. DNA is an extremely long molecule. To fit it into the nucleus, the cells form nucleosomes. Within the nucleus, there are proteins called as histones. DNA is wrapped around these histone proteins, specifically 8 histones, and an additional histone stabilizes the structure. Each structure is called as a nucleosome, and they are later on packaged together to form chromosomes. Linker DNA connects nucleosome with the previous and the next nucleosomes. As you can see in the picture, this is the structure of a nucleosome. These nucleosomes later on form chromatin fibers, and then chromatin fibers are shaped into the chromosomes. Hershey-Chase experiment. Before the middle decades of the 20th century, biologists were unsure of the genetic material of living organisms. Some thought it was protein, but some believed that it was DNA. In 1951, two researchers, Alfred Hershey and Martha Chase, confirmed DNA as the genetic material. In their experiment, they used radioactive isotope labeling. In this setup, the particles of isotopes are released during their decay, which allows radioisotopes to be detected. Two bacteriophage viruses were labeled with sulfur-35 and phosphorus-32 isotopes. Sulfur-35 indicated protein as the genetic material, while phosphorus indicated DNA. After labeling, both bacteriophages infected Bacterium E. coli. As a result, the E. coli infected with phosphorus-32 showed radioactivity, while the E. coli infected with sulfur-35 did not. Since phosphorus is found in DNA and is the indicator of DNA, this result proved that DNA was the universal genetic material. Chargaff's rule. DNA was thought to be a tetranucleotide molecule in repeating set units of 4 nucleotides. Erwin Chargaff did a research to show proportions of nitrogenous bases found in various sources of DNA. He used paper chromatography. In his results, he discovered that adenine number was the same as thymine, while guanine number was the same with cytosine. This data disproved the tetranucleotide theory. And this is the setup of paper chromatography. Exercise. In a sample of DNA, it was found that 18 of the nitrogenous bases were thymine. According to the Chargaff's rule, adenine and thymine number must be the same, which leaves here 18% as well. We have a total of 64% left, And since guanine and cytosine number also should be the same, this leaves them 32% each. Thank you for watching. Have a nice day.